Hi, I am Paco López-Cuevas and this is a scribble video of our recently published story. This work is a collaboration between a chemistry lab and our zebrafish inflammation lab at the University of Bristol. Our goal was to devise potential therapeutics to reprogram the innate immune response, which has remarkable cancer surveillance capacity, to make it better at killing cancer. We reprogrammed these innate immune cells, macrophages in particular, by feeding them miniature artificial protocells containing reprogramming cargoes. In this instance, the cargoes were anti-MUC2-3. And these protocells are injecting them into the bloodstream. In our lab, we use the zebrafish to model cancer because of its genetic tractability and translucency, allowing us to watch how immune cells interact with cancer cells in real time. Macrophages have remarkable surveillance capacities, and so they can detect arising cancer cells. However, what we often see in our cancer fish is that once macrophages find cancer cells, they nurture rather than killing them to help them grow by secreting trophic factors. These cancer-promoting macrophages are typically referred as to anti-inflammatory macrophages. Our aim in this work was to harness this macrophage cancer surveillance and reprogram them towards a pro-inflammatory cancer-killing state. To do this, we used protocells to deliver reprogramming cargoes to macrophages. We chose to load protocells with anti-MUC2-3 cargoes to inhibit macrophage MUC2-3, given that this microRNA was previously associated with poor cancer prognosis, and several studies had reported that MUC2-3 inhibition can drive an increased pro-inflammatory response. Therefore, we tested the anti-cancer potential of anti-MUC2-3 loaded protocells by injecting them in our zebrafish cancer model. First, in zebrafish larvae that had small cancer clusters in their skin, and subsequently in zebrafish adults carrying large melanoma tumors in their tail fins. Cancer growth was diminished in both situations, thanks to the ability of our protocells to effectively reprogram macrophages and enhance their anti-cancer activities. Finally, to test the potential clinical application of the protocells, we used macrophages from humans to culture them in vitro and observed that pro-inflammatory reprogramming was also possible in these human cells, suggesting that this could be a good treatment against human melanoma. This story is now available and can be read in the Advanced Science Journal. Thank you for watching our scribble video.